Hello, beautiful people, and welcome back to day 4 of genetics. So make sure you have your notes for day 4 out. We will be going through this PowerPoint together, so you guys have got everything you need to fill out that worksheet that is due Thursday at midnight. So today's aim is, how does DNA and RNA assist in protein synthesis? Now, if I were you, I would annotate 3 different things. First, I would annotate DNA, and we know that that is double-stranded. This does not want me to write. Double strand. I would also annotate RNA, and we know that as opposed to DNA, RNA is single-stranded. And finally, I would underline protein. And we know that proteins are made up of amino acids. And all 3 of those things will come in handy today. So make sure you annotate that on your paper. All right, now today's do now. The first question is, if 15% of a DNA sample is made up of thymine, abbreviated T, what percentage of the sample is made up of cytosine, abbreviated C? So I'm going to show you guys how to do this. We know that DNA is made up of 4 nucleic bases. And those bases are A, which always pairs with T, and G, which always pairs with C. So in this problem, we are told that 15% of that DNA is T. So next to T, I want you guys to write 15%. Now we know that T always pairs up with A. So if we have 15% of thymine, we're also going to have 15% of adenine. Now, I hope you guys can add. Now, if we add 15 plus 15 together, what does that equal? I hope that you said 30. If not, we need to talk. Now, if 30% of our DNA is made up of adenine and thymine, that means we still have 70% of our DNA left. Because, oh, now I'm messing up. 100% minus 30 is 70. So we still have 70% of our DNA to account for. And that 70% is going to consist of guanine and cytosine. So I'm going to let you guys figure out what the correct answer is. Yeah, come back to Thursday's class with your answer ready. All right, number 2. Chromosomes can be described as blank. So I will draw you a picture. Keep in mind, this is what your chromosome looks like. And that chromosome is made up of a bunch of different coiled structures. And if we zoom into that coiled structure, it's going to look something like this. Now, I'm clearly no artist, but I want you guys to use that image to help you figure out what the answer to number 2 is as well. So once again, come to Thursday's lesson with your answers for number 1 and number 2, and be ready to discuss those answers with the class. And moving on. All right, I'm going to read through this passage, and then we are going to answer questions 1, 2, and 3. So part A, RNA to protein. So far, the genetic information stored as base sequence has moved from the nucleus to the cytoplasm by using RNA. Keep in mind that when we say base sequence, we are talking about A, T, C, and G. Another problem remains: how to use that nucleotide base sequence in the RNA to build a protein with the correct sequence of amino acids. This problem involves a change of language from the base sequence language of RNA to the amino acid language of proteins. This process is called translation, and it occurs in the ribosomes. Built into every living cell in the world is a genetic code. It is called the triplet code. Each different combination of 3 bases makes up a word called a codon. Each codon represents a specific amino acid. Each 20 Each amino acid has at least one codon and most have more than one. I'm illiterate. This genetic code is universal. In other words, all organisms on Earth use the same genetic code. For example, the codon GCA stands for the amino acid alanine in all life forms, from bacteria to trees to humans. This similarity among living things is good evidence that all organisms evolved from a common ancestral life form in Earth's distant past. So as you can see on this screen, I have color-coded each of these paragraphs. I want you guys to use the information that I've color-coded to answer questions 1 through 3 on your own. I will give you a hint. Number 1, use the information in blue. Number 2, use the information in purple. And number 3, use the information in red. Okay, finish those on your own and bring those answers with you on Thursday. So now we're going to go through some animations together of what this looks like. So we're trying to answer the question, how does mRNA code for proteins? So let's do it. So first of all, we know that mRNA has to leave the nucleus. And when it leaves the nucleus, it goes to the cytoplasm. It goes to the ribosomes in the cytoplasm. And it does that because we know that ribosomes are what actually create proteins. So proteins are built from the instructions that we find in mRNA. Okay, so here we have a picture of our mRNA sequence. And then right here, we have a picture of amino acid sequences. Now we know that amino acid sequences, when they come together, they build up proteins. But the big question is, how does this happen? So how do we go from this amino acids, or I'm really struggling. How do we go from this mRNA sequence to having this amino acid sequence that builds up proteins? So today we're going to figure it out. All right, so here we have a DNA sequence. As you can see, those are made up of A's, T's, C's, and G's. However, we talked about yesterday a process called transcription. And that process of transcription is when DNA, let's write down transcription. Transcription is when DNA is turned into an mRNA sequence. And you'll notice that one of the big differences here between DNA and RNA is that we no longer use T's, but we use U's instead. Okay, so this process is called transcription. If you've heard of the word transcribe before, transcribe, it means to write something down. So essentially, this DNA code is writing down that mRNA sequence. We're writing down that code that we're going to need to later build proteins. Okay, so this is called transcription. Now, after it has been transcribed, that mRNA sequence is read by ribosomes. And now this is the big process we're trying to uncover today. So somehow that ribosome is able to read that mRNA sequence and turn it into proteins. And I'll go ahead and tell you that this process right here, from mRNA to protein, is called translation. And when we talk about translating something, we're essentially making sense of it. So somehow, this ribosome is able to read the mRNA sequence and turn it into protein. And that process is called translation. So then after the mRNA sequence is read, we create amino acids. And as I've told you a million times by now, we know that when these amino acid sequences come together, they build up protein. But one question that we're trying to figure out is how can we code for 20 amino acids when we only have 4 DNA bases? So these are our amino acid sequences right here. There are a lot of them. There are 20. And yet we only have A's, T's, C's, and G's. So we're trying to figure out how do those 4 letters create proteins? Okay, so we know that mRNA codes for proteins in triplets. Now, we talked about this yesterday, but when we talk about triplets, we're actually talking about the word codons. Okay, so first up, we have a DNA sequence, and then we know, there goes my ribosome. We know that when we go from DNA to mRNA, all of those T's are suddenly U's, and then those U's allow the ribosome to read that sequence. So the ribosome comes across, and once again, when we go from DNA to mRNA, that is known as transcription. Okay, now I'm going to split our mRNA sequence into codons. Remember, we've got them in triplets, so they're divided by 3. And now this is where the magic happens. Okay, so those codons are able to specifically code for a type of amino acid. Okay, so I'm going to show you what that looks like. Here we've got it color coded, where each of these codons is actually, it's a code for a different type of amino acid. Okay, so now each of these series of 3 is known as a codon. Siri,
Okay,
I don't want to
I found this on the web for series of 3 is known as a codon.
Siri, I don't want to talk to you. Where was I? So a codon, as we know, is a block of 3 mRNA bases. That will come up several times, so make sure you guys are comfortable with that definition. So whenever we split the mRNA sequence into codes or series of 3 or triplets, that is a codon. All right, now, I know this chart looks super confusing, but I promise it's not as bad as you think. So this is the mRNA code, and it is used for all life. That means if a thing is living, it uses this mRNA code. Now, the fact that all living organisms use this same sequence of code is a really strong indicator that we all have some sort of common origin for life. So this is a huge bit of evidence that's used to support evolutionary theories because everything that's living uses this. Now, one cool thing is this code actually has duplicates inserted, meaning that there are several codons for each amino acid. And this is almost like mutation insurance, meaning that if something happens and the mRNA code messes up a little bit, there are safeties built into this code that protect you from any damage. So for example, you can see here that these 4 sequences all code for the same amino acid called leucine. So the first 2 letters are the same for all of them, C, U. But this mutation insurance comes in the fact that although we have a different last letter for each of these, they all code for leucine. So if while your transcription and translation are happening and something happens, mutation happens where a U is accidentally coded instead of a C, no worries, it's still going to result in leucine. The same thing happens over here with proline. Okay, the first 2 letters for all of these mRNA sequences are the same. They all start with CC. However, if a mutation occurs where the last letter of this sequence is changed, either from U to C or from A to G, it doesn't matter because all of it is going to code for the same amino acid, proline. So this happens, or this is built into the code so that just in case something happens and mutations occur, it doesn't change your amino acid sequence. Okay. Now, this is something important you need to know. The start codon is AUG. That means that anytime your cells or the mechanisms inside your cells see the codon AUG, that means that it knows it needs to start translation. Okay, so this codes for the amino acid methionine, but anytime you see AUG, that means start transcription and translation. On the opposite side, anytime your cells see these stop codons, whether it be UAA, UGA, or UAG, the cells know, okay, we need to stop transcription and translation, we're done. Okay, so make sure you make note of those. Your start codon is, Your start codon is AUG, and your stop codons are UGA, UAA, and UAG. All right. If you guys have any questions about all of this, please write down your questions and be sure to ask them in tomorrow's live chat. Okay. All right, so how are the codons matched to amino acids? Once again, we here have our DNA sequence. We also have our corresponding mRNA sequence. We've talked about how to match up these base pairs for the past 2 days. We know that with DNA, A always pairs with T and C always pairs with G. And with RNA, it looks a little bit different. We have A pairing up with U and G pairing up with C. So you can see that in this sequence right here. And they are separated into sections of 3 because that represents our codons. But here's a super cool thing. Okay, now we have this molecule called tRNA, and that stands for transfer RNA. So as soon as the mRNA sequence is made, remember that mRNA stands for messenger, tRNA recognizes those codons, and it's like, okay, it's my time to shine, here I come. And what it does is it actually comes in with the amino acid tucked on its back. And it knows how to match up with that mRNA sequence because it, as you can see, matches up with the opposite pairs as well. So because this is a type of RNA, you see A, U, C, and G being used. So here, the very first codon is AUG. So tRNA knows to come in and match up with it with the sequence UAC. And because it has UAC, it's carrying the amino acid MET, which stands for methionine or methionine. So tRNA is going to do that for every single sequence of this mRNA strand. And every single one of these RNA sequences or these codons is carrying a different amino acid. Now, tRNA's codons actually have a fancy word. We call them anticodons. And we call them that because they are essentially the opposite of the original codons. So whenever you see the word anticodon, we're talking about tRNA. Because the anticodons right here match up with the original codons on the mRNA sequence. Okay, so tRNA will do this for every single one of these codons found in the mRNA sequence, and when it matches up, it drops off these amino acid sequences. And of course, when we have the ending amino acid sequence, what are we going to have? Proteins. Let's keep going. So an anticodon, please write this down, is a block of 3 tRNA bases. So once again, when we talk about anticodons, we are talking about tRNA. All right. Now, I've told you guys that when we turn mRNA to protein, we're talking about translation. I think these are notes you have to fill in on your paper, but don't quote me on that. So we're now going to talk about each of the components that make up translation. So what's known as the working instructions is mRNA. Remember that this mRNA sequence was originally transcribed, meaning that it was written down. So it's kind of like the instructions manual for what's about to happen. So mRNA is the instructions, and that is what that looks like. The reader, so what is doing the translating, is the ribosomes, because the ribosome is able to read those instructions, make sense of it, and then figure out what needs to happen next. So then here's the ribosome going along the mRNA sequence and reading those instructions. And finally, the transporter is tRNA, or transfer RNA. Remember that when the ribosome reads that mRNA sequence, tRNA then comes in with the amino acids and just plops those amino acid sequences down to eventually make all of these amino acids come together to build a protein. Okay. So here, this is just showing you how mRNA is eventually turned into protein. Oh, there's one more. All right, now this is summarizing everything we've talked about so far. So, starting with the original gene all the way to protein. Here we have DNA inside the nucleus of our cell. But we know that DNA can't leave the nucleus, so we have to turn that DNA sequence into mRNA. Remember, the single-stranded copy of that original DNA. And when we go from DNA to RNA, remember that process is called, oh, what did I do? What's happening? I'm so glad you guys are here watching me struggle. Okay, once again, when we go from DNA to RNA, that is known as transcription, because we are essentially writing down the instructions that the ribosomes need into that single-stranded form. Okay, so that is known as transcription when we go from DNA to mRNA. So here's our mRNA sequence. Oh, it's written. Now the next step is we need to make mRNA turn into protein. So we then bring in the help of our ribosomes. Ribosomes read through that sequence of mRNA, and this is known as translation because the ribosome is able to translate that mRNA sequence into appropriate proteins. So then after the ribosome reads that code, the tRNA comes in. Where's my tRNA? There it is. It comes in, it matches up with those codons, and it drops off amino acids. So then when the amino acid sequences build together and form a chain like this, they eventually form a protein. And now, when those proteins build together, they essentially, hold on. So when the proteins are made, that is what codes for your traits. So things like eye color, things like hair color, etc., are all coded for by these proteins. So proteins make you who you are. Okay, and this is just another summary of what we just talked about. So we know that in the beginning, DNA is found within the nucleus of the cell. Remember that DNA is the blueprint of what makes us human. Okay, so then DNA is transcribed into mRNA through transcription. That mRNA then leaves the nucleus and goes to the ribosomes, which is known as the protein factory. That's known as translation. Then tRNA drops off the amino acids. The amino acids build a long chain. And once they finish building that long chain, we have proteins. And then we know that finally, proteins code for traits, which is what makes you which is what makes you live, which is what gives life to this little human. Oh my God, that's not a human, that's a penguin. But these traits are what give the protein. You guys are watching my demise on this live recording. Send help. The traits are what make this penguin have its characteristics and express different traits. That was a struggle. All right, now there are two videos that I'm going to attach to this video that I want you guys to watch. One of those videos describes what is a gene, and the other video is talking about DNA. After you guys watch those videos, you guys are going to essentially write a tiny little essay. I don't want to say an essay because it's kind of a paragraph, but you're going to write a paragraph describing the process of protein synthesis. Now you must include the following words in your paragraph: tRNA, mRNA, codon, amino acid, protein, DNA, gene, translation, transcription, nucleus, cytoplasm, and ribosome. So that seems like a lot, but we have given you all of the information you need to write that paragraph. Okay, now I'm going to have the videos playing at the end of this video if you would like to keep watching, or I'm going to embed the YouTube links as well so you can watch them there as well. So you've got two options. You can either keep watching this video where I will embed the videos, or you can exit out of this and go to YouTube and watch the same videos there. So thank you. That is day 4. I'm sorry you witnessed me having a stroke on air. But if you have any questions about what we just talked about, please write them down and I will answer them tomorrow in class. Until then, Have a great day. I am now going to turn off my recording, but I will have the videos playing at the end of this segment. So goodbye, and I'll see you guys tomorrow. Let's see. There we go.
Temper tantrums. Stated Clearly presents, what exactly is a gene? Each one of our cells contains 46 strands of DNA. A single strand is made of millions of particles called nucleotides, and these nucleotides come in 4 different types, which scientists have labeled A, C, T, and G. A gene is a special stretch of DNA, a sequence of A's, C's, T's, and G's that code for something. A gene contains information for a cell to read and use, but what exactly does that information do? You might have heard that there's a blue-eyed gene, a freckle gene, possibly even an anger gene, but single genes don't literally make things like eyeballs or freckles or temper tantrums. Genes make proteins. Those proteins then interact with each other and all sorts of chemicals inside the body to build things like eye pigments, freckles, and mood-altering hormones. A single strand of DNA contains thousands of genes or unique protein recipes. Humans have roughly 20,000 altogether. Some genes are small, only about 300 letters long. Others are well over 1 million. The length and sequence of a gene determine the size and shape of the protein it builds. The size and shape of the protein determine the function that protein will have inside the body. Hemoglobin, for example, is a protein structure found in red blood cells. Its unique shape and size allow it to capture oxygen molecules when blood flows near the lungs, and then release them later when blood flows near oxygen-starved tissues. Pepsin is a digestive protein. Its unique shape allows it to break down food inside your stomach so it can be absorbed by the body. Keratin is a structural protein. Its unique shape and size allow it to link together with other keratin proteins to form hard structures like fingernails, claws, and beaks. Different creatures have different genes, which is ultimately why their bodies look and function differently. But one of the many reasons scientists believe all life on Earth is related is that the basic DNA code, the language of A's, C's, T's, and G's, is pretty much the same for all living things. Many creatures even share some of the same genes. You might not be too surprised to learn that humans and chimps, which are closely related, share 96% of their genetic code. But what would you think a lowly fruit fly has in common with a beautiful swimsuit model? Surprisingly, about half of its genes. Because all creatures use DNA in pretty much the same way, genetic engineers have found that if they take a gene from, say, a bacteria cell and insert it into the DNA of an animal or a plant cell, that animal or plant cell will then read the new gene and produce the bacterial protein. Engineers have mixed and matched the genes of different organisms to produce many new creatures, including corn that is toxic to insects but supposedly safe for human consumption, tomatoes that last up to twice as long in the grocery store before going bad, and a new form of bacteria that produce the human protein insulin, which we then collect from these bacteria and give to people with diabetes who need extra insulin to survive. So just to sum things up a bit, what exactly is a gene? A gene is a special stretch of DNA, not the entire strand of DNA, just a segment that codes for something. Each gene is like a unique recipe, which usually tells a cell how to make a protein or a group of proteins. Different creatures have different genes, but all genes are written in the same basic DNA language of A's, C's, T's, and G's. I'm John Perry, and that's genes stated clearly. Stated Clearly presents, what is DNA and how does it work? DNA, also known as deoxyribonucleic acid, is a molecule. It's a bunch of atoms stuck together. In the case of DNA, these atoms combine to form the shape of a long, spiraling ladder, sort of like this one here. If you ever studied biology or saw the movie Jurassic Park, you probably heard that DNA acts as a blueprint or a recipe for a living thing. But how? How on earth can a mere molecule act as a blueprint for something as complex and wonderful as a tree, a dog, or a dinosaur? To help answer that question, let's first take a quick look at amino acids. Amino acids are tiny little chemicals inside our bodies that are so important, they're often referred to as the building blocks of life. There's about 20 different kinds, each with their own unique shape. The neat thing about them is they can be attached to each other, kind of like Legos, to produce an endless variety of larger particles known as proteins. Amino acids make up proteins. Proteins, along with other chemicals, combine to form living cells. Cells make up tissues. Tissues make up organs. And organs, when they're all put together and functioning, of course, combine to form living creatures like you and me. These proteins that make up our bodies, and keep in mind, there's millions of different kinds of proteins. They each have to be formed in the perfect shape in order to function. If they're the wrong shape, they usually won't work. That's where DNA comes in. DNA does a lot of interesting things, some of which we don't fully understand, but one of its main and most well understood functions is to tell amino acids how to line up and form themselves into the perfect protein shapes. In theory, if the right proteins are built at the right time and in the right place, everything else from cells to organs to entire creatures will come out just fine. This here is a simplified model of DNA. It shows us that the steps of the ladder are made up of 4 different kinds of chemicals shown here by different colors and letters. If you look at just one half of the molecule, you can read its chemical sequence or genetic code from top to bottom, sort of like a book. A single strand of DNA is extremely long, millions of letters long. It spends most of its life coiled up like a noodle, living inside the nucleus or the centerpiece of a cell. Amino acids, however, live outside the nucleus in what's called the cytoplasm. To help DNA interact with the cytoplasm and convert those amino acids into proteins, special chemicals inside the nucleus make partial copies of the DNA code. These partial copies, called RNA, look a lot like DNA, but they're shorter, of course, and they're missing one of their sides. Their small shape and size allows them to fit through tiny pores in the nucleus, out to the cytoplasm, and into the mouth of another particle called a ribosome. Ribosomes are protein-building machines. They read the RNA code 3 letters at a time, suck amino acids out of their surroundings, and stick them together in a chain according to the RNA code. As the chain grows, it bends, it folds, and it sticks to itself to form a perfectly shaped protein. Every 3 letters of the RNA code tell the ribosome which of the 20 different kinds of amino acids should be added next. For example, CAA tells the ribosome to grab a glutamine. AGU tells it to grab a serine, and so on. Once a protein is built, It can then go on to do a number of different things, one of which could be to help form a brand new cell. So to answer the original question, what is DNA? DNA is a molecular blueprint for a living thing. How does it work? DNA creates RNA, RNA creates protein, proteins go on to form life. This entire process, as complicated, as sophisticated, as magical as it might seem, is entirely based in chemistry. It can be studied, it can be understood. I'm John Perry, and that's DNA stated clearly.
Now, if you've made it this far,
Thanks
here's
for
a
watching. If
picture
you
of
liked
my
what you saw,
cat
make sure to
Connor.
give us a thumbs up on YouTube
He
and
wanted
share
to
it
say
with your
hello.
friends on Facebook or Twitter
Okay, bye guys, see
or
you
wherever
tomorrow.
it is you happen to share stuff with your friends. Subscribe to us on YouTube if you want to see more videos. If you have any questions or comments, you can post those in the comment section on YouTube. We do read those, by the way.